Welcome to Bio class. Today we will discuss about cancer. The cancer, this word came from Latin word cancrum, which means crab. Greek word carcinoma, also in English, used in English as carcinoma, means cancer. Okay. Cancer actually the abnormal and uncontrolled division of cell that invade and destroy the surrounding tissues. Okay, that is a definition. Cancer, it starts when it starts in a normal cell, begins to grow and divide and uncontrolled, and it's formed this tumor, the cluster of mass of cell tumor, which is called neoplasm, malignant cells, malignant tumors, many names. And these tumors can be the different type of shapes and size you can see here. But one thing you have to remember, that all the tumors are not cancer. So tumors means does not mean every tumor is cancerous. The tumor which not caused cancer, known as benign tumor. So let us know about the benign tumor and the malignant tumor. What is the basic difference? So benign tumor which is not fatal, it does not give rise to cancer, and the malignant tumor which causes cancer. That is the first difference. Other difference are the rate of growth. Here it is slow, but it is very fast. Here the spreading it remains restricted, does not spread, but it spreads rapidly. Metastasis, afterward I will discuss here. It does not occur, but here it occurs, and here it is a latent stage. It is absent, here it is present, and risk factor it is told already. Now come here and metastasis. Metastasis, what is it? Some cancer cells penetrate blood and the lymph vessel and spread to the distinct organ where they are established and new cancer colonies. This moving and settling process is known as metastasis. See that, SIS, and the distant colonies are known as metastasis, SES. So it means that metastasis it moves and it goes in different areas. So this tumor, it invade inside the bloodstreams and it spread the different areas, which is called metastasis. So it is found only in malignant tumor. We know that the cancer is one of the dangerous disease nowadays, and often we are asking about the cancer, that the people are saying that they are in, the patient is in certain stage, I mean third stage or fourth stage, and everybody, I mean we are talking about it, that fourth stage, it means dangerous stage, and third stage or second stage. So let us know about the staging little bit, maybe not in a book, but you should know that. Cancer staging, how the staging it is done. The size of the tumor, whether it has invaded an adjacent organ, how deeply it has penetrated in the lymph node, and whether is the spread, the distinct organ, the metastasis taken place or not. So this is called TNM staging systems. T stand for tumor, forms the tumor. N stand for node, it means that in lymph node it invaded. and the metastasis, it means that the metastasis taking places or not. So, this is TNM staging. If we see this one little bit in this, there are there are two stages. I mean, clinical examination, I mean physical, and the pathological examination. So, pathological tests. So, stage 0, it means that the carcinoma in situ, it means the tumor in its place. Stage 1, here the cancer are localized. Second stage, cancer are ready to invade, and third stage, here it affects lymph node, invade lymph node, and the fourth stage, the metastasis takes place and spread and other organ, it is done. Now, if we see this part here, that it is written like this kind of formula. Suppose C, if it is written, then C stand for the clinical examination. T3, here it stand for the tumor affects the lymph node. Invade N1, if it is written, so that means the node, it is stage 1, the lymph node affected is localized, and M0, if it is then metastasis, no metastasis, it is there. Okay. So, in this type of formula it is used. In case pathological, if it is like this, then when it is lymph node, it is 0. So, metastasis is no need to write, like this type of it is written. Now, very big question here, that what is the causes of cancer, the how any cell or any individual they affected by the cancer. We know that several I mean carcinogen it is available, like benzopyrene, tar, nicotine, tuxaphene, ultraviolet ray, saccharine, halogenated water, many other that can cause the cancer. In cigarette, that benzopyrene it is there, that it also induced. But these are the carcinogen. What is the internal way and process here, that basically here in your standard, that I can discuss these two things, that protooncogene and the tumor suppressor gene. Protooncogene, it means that it is a normal gene which is available in every individual's body. Protooncogene, when it's mutated into oncogene, always remember from the non-functional to the functional gene, the oncogene, then the cancer, the disease will start. That is why the cancer is also known as, I mean, the study of cancer is known as oncology. The Greek word onco means tumor. Tumor suppressor gene is always active in our body. Always active in our body. Anyhow, in case of mutation, it is mutated by that benzopyrene, tar, nicotine, that carcinogen, UV ray, saccharine, all this. So, if they are inactivated anyhow, so that cancer can causes. So, this is quite amazing, that one gene is becomes active, so it causes cancer, another is inactive, then it causes the cancer. Let us see this. Here oncogene, when it converts into oncogene, it gains the function, the mutation in protooncogene, and tumor suppression gene is the loss the function of the mutation in the tumor suppression gene. Let's we can see this to understand this fact. In tumor suppressor gene, Look at here, it acts in the two ways, as a gatekeeper, as a caretaker. Okay. Gatekeeper means it regulates the cell cycle. Cell cycle, if this cycle, if it moves, see the arrow here, and here it acts like a gatekeeper. It means that when it is valid, it can allow and it can stop. So one regulation is there. Checkpoint, it can controls, and caretaker is it can repair the DNA also, the minor damage it can repair. Okay. So one thing you can able to understand that if any DNA is damaged, in cell during cell division, the cell cycle, it can be repaired. Number one, it can be if it is not repaired, then it is destroyed. I mean, whole cell it is destroyed, so that that kind of things will not copied with the autopsies, and it can be seized for the some time, that it can get some time to repair. So all these things are there. We will take one example and we will explain all this. So one tumor suppressor gene is P53. P53, it is known as ultimate controller of the DNA damage. It is called guardian of the DNA. Many names are there. The protein which weight is 53 kilodalton, that is why name so P53. P53, it is found in the gene in normal cell, and it is active when the DNA is damaged, and in that kind of problem is there. Otherwise also it is in moderately active. Now before that, we have to know about the cell cycle. We know that cell cycle, here the synthesis, I mean DNA synthesis, it takes place after the G1 phase. Suppose just after the divisions, cell divisions, telophase, we know that here the chromatid all again it turns into chromosome, and here any problem or any mistake is there, anything torn or any DNA is damaged, so that part, when it happens, then P53 will be activated. In normal cases, look at here, this is the checkpoint, and here is the cyclin and the cyclin dependent kinase CDK, both are available. In normal case, both are functional. Both are functional in normal cases. Okay. When it is functional, when it is functional, then it allow that from G1 to S phase. S phase, so cell division will normal, it will go, and cell can divide in normal rhythm. But in case of DNA damage, what will happen? The P53 gene will become active, and it forms that P53 protein, that it is actually transcription regulator, and it activates several, I mean other gene, other mRNAs, and forms that P21 protein. P21 protein here, P21 protein will block, it will block this area, the P21 will block this, okay, cysteine CDK, and it is emergency, and immediately the cell division process will cease, and it becomes non-functional, and it is ceased. So that the damage copy is not copied inside the synthesis phase, I mean protein synthesis. Here it can possible that the DNA will get the time to repair the damage, or if it is not possible, then it goes for the cell death apoptosis. If one cell will die in that so many cells when divided, then nothing will happen, but the body will be saved from the any kind of genetic type of problem. Now just imagine when this things is not there in your body and it is mutated, so it will not work, so all this work will not happen. So what will be there, that G1, this will be I mean with the defective things it will run, and in that way the cell regulation will lose their I mean control, and huge cell mass will be there, and that is the malignant tumor. Okay. So P53, about P53 I am just telling you, it is one recessive gene also can do work. It means that in chromosome it is found in, suppose in these two areas. So in both P53 is there. If one is mutated, another can do its normal work, like heterozygous form. Heterozygous form it can do its work, but when both are losing their function, then the huge problem will be there and it causes the cancer. Okay. Another side, the proto-oncogene is converts into oncogene, and here it effect on the growth factors, and so that a cell loses its regulation of the growth, and huge number of cell will grow. So that is the it forms the mass of tumor. So that is about the cancer. Diagnosis also, we know the biopsy, the study of the blood sample, CT scan, MRI, microscopic examination of the body, X-ray or fluorescence, all this it is there. So still that in treatment we are little bit improved, but it is non-curable in most of the cases. The chemotherapy, radiotherapy, immunotherapy, even the digital database also is applied in this. Okay. So that is about cancer. Thank you.